Yeah. All right. So, great. That would be good. Hello, my name is Jordan Clemsen, and today I'll be discussing the paper that's published in Translational Psychiatry. That was structured microbiome inflammation with brain access and medicated bipolar depression type 2. First of all, I'm going to go over some of the background information because this is a very complex, multifaceted paper with perhaps too many different angles that they're trying to study. But first, does anybody know what the central dogma of molecular biology is? So I don't have to repeat it at all.
Oh, do repeat it.
I'll go over it anyway. So transcription goes from DNA. For these four base pairs are essentially the coding blocks of our whole entire genome. Whether it's DNA or RNA, you have adenosine, thymine, guanine, and cytosine in your DNA, and that usually is transcribed into RNA, which is the following base pairs with the same nucleotides except for uracil of thymine, and from that transcript into the ribosome, you make a template copy, as you can see here in my nice little image. It's color coded, and from there you make proteins. So essentially, that's majority of our bodies and how we code for life and everything like that. There is numerous processes that come from interpreting this data. For instance, the codons that I have listed right here which are codons, by the way, are three clusters essentially of these nucleotides, and these and the combinations of these clusters of the nucleotides make a certain protein, and then in the sequence of those proteins themselves codes for the whole entire macromolecule itself, which then helps, which differentiates each part of the gene itself. So, for instance, the codons that I have listed here, UUG and AUG, are both start codons. That essentially tells the RNA essentially where you should start reading from. I like to think of it conceptually similar to whether we count from 0 or whether we count from 1. And that'll be important later when I discuss essentially how this is altered, I guess. But in summary, the central dogma of biology is essentially we transcribe from the DNA to the ribosome to make messenger RNA, and this messenger RNA is then transcribed by proteins, and that kind of makes up our body.
Can I ask a question about
Yeah.
codons? They feel kind of arbitrary, aren't they? Because if they're like trigrams, but if you shift within the sequence, all codons are different.
It's completely different. So yeah, so there are transcription factors that would do that. Or if it just happens randomly in the body, it's called a frame shift mutation, because we take everything over and we shove it over one, and then all of a sudden it's completely different, right?
So there is a reference point where you start,
Yeah,
and then you're saying, oh, next 3 is a codon. How then
absolutely. So it usually has to do with the start codon, which I have listed down here. So when the DNA, so when the well, depending on what it is, the RNA polymerase, for example, let's just say for that, we'll see the UG or the AUG, and it'll use that as a reference point to start. So that's where it knows to start. So if it is shifted over, then yeah, it could be completely different. Those are called frame shift mutations. However, there is sometimes, depending on the organism, I don't want to go too much into the weeds here, but there's a thing called codon optimization, which some organisms actually do that on purpose just so they can get a more complex code, if that makes sense. But in summary, the main thing I'm trying to get across here was just the transcription, which is just, DNA messenger RNA and messenger RNA for proteins. Sorry. Hopefully that simplifies it. But long story short, yes, it can be a lot of things. It could be a mutation. It could be a strategy for more diverse optimizations. I'm studying virology, and some viruses do that on purpose. Long story short. So,
Yeah, thank you.
yeah, thanks. I hope that answers your question. And another concept that's actually used in this paper too is the interaction of microRNA itself. So microRNA are these little hairpin loop structures that essentially they can block off or stop part of the message from being read itself. So let's say we have a stop codon that's all of a sudden blocked off. It wouldn't the whatever it's reading is the messenger RNA would not be able to tell where it started, and probably wouldn't be the whole entire gene itself because it wouldn't know where to start. So as you can see here from the diagram, how these microRNA work, as they attach to certain messenger RNA, depending on what the gene will be, it either upregulates or downregulates the protein expression itself, which essentially can change a whole bunch of things. It regulates somewhat to a transcription factor. Sometimes it adds a methyl group onto this DNA, and this changes one base pair. And as we previously went over, one of these base pairs, if one of these changes, cool.
That's not good.
Is that like a? Oh, that's me.
Oh, no. Yeah. Go.
Oh, okay. I was like, I did something wrong. Oh no, I broke something. Okay.
Before the fire alarm, we can.
Yeah. So one base pair changes, the whole entire codon changes itself. The codon could be a different amino acid, potentially, and then that changes the whole entire protein itself. So these microRNA are incredibly important because they can change the whole entire message. Essentially, there are genes that actually code microRNA, so you can have microRNA that then code for other microRNA, and it gets more complex from there. There's a whole bunch of these. These are tiny. These are really tiny. They are actually down to the nanometers. So they're about the size of DNA itself, which is so they can get in and out of the cell really easily. So there's quite a lot of these just floating around our bodies, and it should go without saying that the Nobel, just a little bit extraneous information, but the Nobel Prize was awarded for discovery of microRNA. It was discovered back in 1993. We just had the Nobel Prize, I think it was last year. Yeah.
Can you quickly repeat? So,
yeah.
Like from the picture, it's more of like a sheath, like a holder for my
messenger RNA. Yeah.
Yeah, for messenger. But then you say, what is its other function?
Communication between cells because it helps regulate the expression of the protein itself. So if I had like a codon here, and I block, and this image itself codes from adenosine, protein, etc. And if I attached a microRNA that blocked off, let's say the adenosine, then that would be a different protein altogether. If that makes sense. That would be a different amino acid. If that makes sense. Well, it wouldn't be a different. It would block off amino acid and make a different protein. If that helps. So they act more as like a negation, if that makes sense, for when it's paired to. And the reason it's important is because this paper does kind of go over bacteria and how it interacts with it. But we'll get to that in a second. And then one more thing, I'm just going to breeze through the bacteria itself. So bacteria have shown, yeah, I'll just say a second. So there's more than just bacteria in the microbiome itself. But this paper doesn't touch on that, so I'm going to just go over bacteria itself. They have a plasmid, circular RNA, a circular genome, which is used to can be unwound into a single strip, and this gene can be shared between bacteria itself, so they can actually teach each other how to code for genes. They have an SOS response, which is essentially like brute force. It's like everybody just guesses on a test, and then we all turn in the test all together, and depending on whether the test and if there's a huge amount of classmates itself, like 10,000 classmates, then one of those classmates will probably have a really good score just out of sheer chance, and then that classmate will carry the whole class. If that makes sense. That concept is very similar with bacteria because they insert their nucleotides into their ROS response, and their genome while they're doing this, and then they can also share the bacillus itself. Bacteria are capable of forming endospores, which allows them to exist in harmful environments like the gut itself. There's a vast amount of bacteria itself. Most we don't really understand yet, which I will touch on, I think, very way after. Um, and each bacteria itself usually has multiple biochemical pathways of fermentation. Um, Off the top of my head, lactobacillus takes in maltose and outputs hydrogen peroxide, which we can think about that in our gut. Has numerous instances. And the final concept I'm going to go over is the microbiome itself, because again, this paper looks at interacting with the microbiome. The enteric nervous system essentially operates from the colon, anywhere from the ascending colon all the way down to the sigmoid colon itself. This nervous system has pathways between the crypt and the villus of the villi. Well, not the villi, but it will have in between the cells itself. In between these cells in the colon, there is tight junctions, which permeate and allow for dendritic cells to communicate with the inside of the colon itself. This allows bacteria to essentially, 90%, off the top of my head, 90% of the serotonin produced is from the gut, and the majority of that has to do with bacteria that are able to produce similar molecules like serotonin in tandem with the microRNA themselves. They're able to communicate in an extremely complex schema that is oversimplified in this image, and demonstrates that there's communication between the brain through microRNA and protein expression, but also through the neurotransmitters, through the enteric nervous system and a feedback response with the vagus nerve. The benefits of the microbiome itself is the fermentation, depending on what organism is actually taking up the space in the gut. Helps not only aids in digestion and nutrition absorption in the colon, because we don't really absorb a great amount of our food, but also the fermentation and the output does have some beneficial effects to the host organs itself. I mentioned serotonin. There's been other papers that have suggested GABA, and there's currently, I think, histamine is being explored also too. This obviously has a lot of effects in pro-inflammatory cytokines and anti-inflammatory cytokines as well. As previously discussed, lactobacillus does ferments hydrogen peroxide, which is a great deterrent for killing other bacteria that you probably don't want in your gut, because hydrogen peroxide is introduced through radicals. Also, for example, the example of lactobacillus, I like to go with that one because it's really it's really simple, and I like that. But it takes up space in the gut, and that space that it's occupying is something that is occupied by lactobacillus, and not something worse like E. coli or botulism, if that makes sense. Or any other gram-negative bacteria that we don't want. There's been evidence that suggests that we have co-evolved with these bacteria itself. While they do not share our genetic makeup precisely, as we don't replicate for them in meiosis and meiosis. We do pass down bacteria through breastfeeding and vaginal birth canals itself when we were born. So essentially, there is a pathway of people. There's a lineage of these bacteria, depending on the person. And finally, a huge part that I want to say, if all that doesn't make any sense at all, there's the fact that bacteria are able to not only produce similar structures similar to microRNA itself, but they're also able to engulf the host microRNA and communicate. So there is influence on protein expression as well, which will be discussed in this paper. Okay, sorry, that's a lot of the background. I think this paper is really ambitious, and I wanted to simplify it as much as I possibly could. Fun question: if you had a functioning pilus and you could like transfer any of your genes, what would you share and why? Engaging question.
Any gene?
Any genes? I don't know. Well, not polygenic because there had to be one, right? It's a fun question. I'm trying to make it fun.
Do you want like to name the gene? Or?
Oh no, like if you could like just like shoot out like a little strand and I poke Mike over there and just like share my curly hair gene or something. Oh, here it is, yeah, it's curly hair. Like I don't know. I was trying to make it engaging. I guess I failed.
I would do that with like an animal. To create a hybrid.
You'll share your gene with an animal, or you will adopt a gene from an animal.
I will share it with an animal first and see how that ends up going.
Give myself cat ears.
Yeah, the lemur too.
Gills.
Okay.
Gills are sick.
Fair.
It's not one gene problem. But
yeah, I know, but it was intent. It was trying to make it fun.
Bacteria are having it easy, right?
Yeah, because like they're well, and when I get to the critique slide, that kind of gets down to that I kind of go into that a little bit more. That when you get to a simpler organism, speciation kind of differs. The difference between me and a zebra versus another bacteria and another bacteria, because they're so simple, one change could be huge, and it also can be shifted out really easily. So, but I'm getting ahead of myself. That would be later. So finally, I'm going to get into the study now that because this paper tries to go into a lot of different things. So why did they look into this? The motivation. So first of all, bipolar disorder 2 symptoms. They suggest that there is a hypomanic depression, which is shared with depression, and there is previous literature that suggests that the microbiome, as previously discussed, alters serotonin levels, dopamine levels, and inflammation in depression. And so exploring in hypomanic depression would be is also interesting and has been really researched, really interesting too, and really hot, I think, in the last decade. There's some of this literature has demonstrated that there's a reduced dynamic amplitude, low frequency fluctuation patterns in fMRI with patients with bipolar type 2, and there have been alterations in the dynamic functional network connectivity with bipolar type 2 as well, and so. And just due to the ambiguous nature of the disease and the misdiagnosis, there's always going to be outliers, which have raised many concerns on top of the already vast, complicated question of the microbiome itself. There's also been a connection between pro-inflammation and bipolar type 2 and pro-inflammatory biota in the gut, not just bacteria, but these also sometimes lead to an increase in pro-inflammatory cytokines. And so being able to distinctly measure that has been crucial to symptomology and hypomanic depression. There's also been previous literature that has shown that the default mode network connectivity is correlated to the microbiota itself. though debatable, which I'll get into later. But despite all of this exhausting research that's really been done on hypomanic depression, there has not been really any uh, tests or studies that have looked into unmedicated changes of the biota. And so this study thought that'd be interesting to explore, which makes sense. So the first thing they did is they had 65 participants, one with medicated bipolar depression, taken from a psychiatric hospital, and with the same number of controls. These individuals were selected based on meeting the DSM-5 criteria and numerous other qualifiers. The DSM criteria qualifies a hypomanic episode that lasts, oh my gosh, I think like 6 or 9 months, I think. If it's longer than that, then I think it kind of like gets arbitrary. But I mean, that alone is kind of weird. I think there has to be a hospitalization in the DSM criteria itself. I wouldn't quote me on this, but I can provide more detail on that and actually bring up my copy of the DSM if need be. These individuals also scored greater than 21 on the Hamilton diagnostic rating system, or Hamilton depression rating system. Sorry, that's not the. There's a couple measures that they use that cut them out. There's one that they use with anxiety as well. The Hamilton depression rating system itself essentially uses a Likert scale for depression, and it tallies up the score. And if you had more than 21, which would be pretty high, then you would be classified as severely depressed. And that was one of the criteria they used. They used multiple measures for each one of these. They used multiple measurements for each one of these measures, I should say. They also had the young manic Racine scale itself, which is this one is a multiple choice survey, which is used for people to assess whether they're individuals of 18 or younger who have manic experiencing mania and a symptom there, which is it's kind of a symptom of depression, but it's kind of the opposite end of depression. I don't want to go too much into that. Each subject was then scheduled from the psychiatric hospital, was evaluated for 3T and MRI. They used frequency fMRI. I don't know why they put that. Blood samples were taken for cytokine levels, and they also did the fecal samples collected too for analysis of the microbiota. So what they did for the imaging itself was they looked at the whole entire time series. They applied the fast form transform. They of course smoothed the voxels itself. They essentially looked at they used the root of the power of AR or G, smoothed to, as I said, sorry, they smoothed to 6 millimeters kernels, and they formed a static functional network static functional network connectivity from clusters of the ALP itself, as I previously described. There's a Pearson correlation with the mean time of voxels, and a correlation map with the z-scale after cluster itself. I had smoothed it here twice. Oh, then they smoothed it again. I forgot about that. They did smooth it afterwards twice, which is an issue with that I will bring up in my critique slide finally. And they did use BFNC as well. They used a sliding window. They said they used 50, but then oddly enough, they said that they validated it afterwards with a 30 window and a 70 window, which I kind of would think, why not just say that in the first place? Why would you say you 50 and then validate afterwards? That's what they use for the imaging. I'll get to the results afterwards for the microbiology component of the paper itself. They measured a whole myriad of cytokines. I want to point out that some of these are anti-inflammatory, some of these are pro-inflammatory. Tumor necrosis factor alpha, depending on the pathway that it goes through, can be anti-inflammatory or pro-inflammatory. Usually it's pro-inflammatory. Interleukin 10 is a pretty normal anti-inflammatory one, and so forth. They used a MagPure stool sample kit for the DNA of each patient. They used PCR for validation. with each one of the 16 RNA. That should be microRNA. MicroRNA illuminating protocol, which they used for microRNA-seq, which is essentially just a clustering method that they use to cluster up the microRNA itself, so they can tell which bacteria are which based on deep learning itself. They as which described as a short RNA sequence. They mapped out each one of the bacteria themselves. They applied a Shannon index for variance. Usually, I've seen in other fields they use ANOVA. This one used Shannon for entropy and the Simpsons index. They also applied a principal coordinate principal component principal coordinate system itself, and of course ran it through a single vector what's it called, a single vector it's SVS, I think it is. I can't remember the algorithm itself, but essentially it's they take it into higher order mathematics. I'll remember the exact. I can share that in a bit. Sorry about that.
Are you doing about classification?
Pardon?
Classification or?
I was talking about how they so they basically what they did is they took it and they made it a tensor so they can apply it to a higher order mathematics. If that helps, which is part of my critiques. But I'll get to in a second when I get to the statistics if that helps. They essentially what I'm trying to say is that they use a different theory and then they use the software LEFSE and essentially try to take it and then, so they can make a model out of all this a multi-model data. If that makes sense. So some of the statistics and then they also they used a chi-square test along with an independent single one-tailed sample and Manhattan-Whitney test itself, so they could differentiate between each one of the Can I pause this so I can go to the restroom real quick?
Yes.
Okay, sorry. Oh, that's better. Okay, so they looked at independent t-test itself. They compared that with the Manhattan-Whitney. Essentially, they used a lot of different mathematics. They used Gaussian randomized field theory to apply a model to. Essentially, essentially what they did is a whole bunch of different t-tests and a lot of data itself. But to summarize it all, what they did is they took this each of one of these data measures and took it into higher order mathematics, and somehow found out that there was a 80% prediction rate for all this data from the resting state the resting state development networks itself in comparison with the microbiota, which I think is kind of interesting. They also found that there was an increase in pro and anti-inflammatory cytokines with the bipolar disorder itself.
Hey, Jordan, I had a question on the side.
Yes, sir.
So, for the fMRI, what did they use for those features? They used the functional connectivity and the balance
So
data?
they use the alpha itself. They use functional network connectivity. They used dynamic functional network connectivity, and that was essentially just it.
Okay.
Which is
How did they determine the regions? Is it like a regional based functional connectivity or seed based?
they didn't really say that. They did mention some regions. They mentioned the what was it? So they mentioned the it was the medial temporal succulus. I think they mentioned. They also mentioned they didn't mention the prefrontal cortex, which is kind of interesting. But they did mention some specific features. They even listed them off in a very detailed, superfluous manner, similar to this. So I don't know. Is the long story short. They didn't. Yeah. Any other questions? Sorry.
So what they classify?
Oh, what were they classified? So they classified the regions of the brain. They also classified the different measurements from the DNA from the fecal samples itself, so they could classify. So because of speciation gets really complicated when you get down to a macro a simple organism or organismic scale. Usually people use RNA-seq. I've been in fields just because it's less arbitrary and like, avoid some of the phylogenetic issues that you run into if otherwise. When you get to a macro scale, you have like zebras, people. It's very easy to tell the difference. When you get to a bacteria itself,
You think so?
yeah. When you get to bacteria, you can't tell if they're pills, if they're sharing with what part they're sharing, what part they're not sharing. And because it's so simple already, just sharing that can be very confusing really quick. So essentially they use RNA-seq, which is just really deep learning plus it's the same algorithm we use here, but it's just applied to the sensory of the nucleotides of the of each collected from fecal samples. So they use that to distinguish which species are which, and then they compare it to a database. I think that was in my previous slide. I breezed over that because that was nauseous, but it was the RDP database itself, so they compare it to that database, and that helps. And then they can determine which bacteria are which, based on that when they include that with the cluster, that helps. Hope I'm explaining that correctly. But yeah, the results were that they reported 80% accuracy for inflammatory cytokines. I hope I answered your question. I have a whole bunch of critiques of this paper because I think it was to say modestly, I think it was I think it was a little over ambitious. I think they tried to do so many different things, and they a bunch of measurements that just kind of ended up making the data more and more messy. And so when they took it into higher order mathematics, it just ended up just being a mess. And I think that's why they ended up with a whole bunch of false positives. They even mentioned earlier that well, they didn't mention, but the prescience of the Young mania scale has already been critiqued for being notoriously high with false positives for diagnosis because of the multiple choice nature of the questions itself. It's really easy to get false positive. These participants were only recruited from 2022 to 2023, so it's only 1 year. And also it's only from one hospital, and there's been previous studies that demonstrate that when you have a when you're in the same environment with the people, like we're probably all sharing microbes right now, so there's a sampling bias in that. And on top of all of that, the sequencing data itself. So due to culturing issues and also numerous other factors, 1% of the bacteria has been measured out of all of the bacteria that we know. So applying that a clustering technique like that really doesn't make much sense. And we don't even have the databases for most of the bacteria out there.
And we probably know the less than 1% of bacteria that are out there.
Yeah, it will. So I think applying that already kind of messed with the nature of the experiment. I think it was going great up until when they applied the mathematics, and then you end up with all statistical noise that just end up with a whole bunch of false positives based on these assumptions. And I think they tried really hard to validate it with multiple different measures, but I think it just adds to the noise itself. The sample size itself, as with controls and the subjects, was only 130 altogether. This is published in Nature, I think. This is where I got it. I was reading Nature, I think, in December, and I thought this could go over it. It's just critiques I had. It also doesn't go into an even more complex virome itself, because not only do we have bacteria that's going around our guts, we also have bacteriophages out there for every basically influencing the amount of bacteria expressed itself. So not only do we have 1%, we also have viruses that are also limiting that 1% as well. There's also a microbiome, which is essentially the fungus itself. This was not measured in the paper at all. It also has, again, an effect on not only the pro-inflammatory cytokines, but also on the type of bacteria expressed. Also, I want to point out that Mr. Bockel's study, that did not include any of the microbiota data, had over a 90% success prediction rate with bipolar itself, just from right out here. So I don't understand how they can get an 80% and we can get better just by cutting it off, unless there's something wrong with the data. I think that other microbiomes would need to be assessed too. And so ultimately, my critiques of this paper is just that there's a lot more that would have to be measured accordingly to get a proper conclusion, besides just nonsense, really. But yeah, that's kind of most of my critiques. I do think that there is room for ICA to try to sort this out a little bit, but I mean, it would really, you really need to sort out a lot of different cofactors and co-founders and mechanisms. But yeah. Is there any questions or any critiques? And then I think if anybody else wanted to share their thoughts on it at all, I'm sorry I'm not feeling the best.
Can I give a quick comment?
Yeah.
Don't be so self-critical. We don't care about you critiquing yourself, we just care about the content.
Thanks. Yeah. Not feeling the best, but wanted to show. Thanks.
Thank you.
The paper is included there. I can include anything else too. There I can include where I got the 1% from as well. There's previous nature studies that go into the culture techniques, if that helps as well.
So is this 1% of the gut biota, or
It's an estimation. This is all bacteria. So because what they did present, they did suggest that there was protobactericides, bacteridates. I can't say that right, but bacteridates have been previously shown to have a positive effect on the quote unquote good bacteria because they help with serotonin expression. Similarly, lactobacillus has also been shown to be a quote unquote good bacteria because it takes up space while also kind of denying the rest of the bacteria with hydrogen peroxide. Those are just a few ones that we know. There's so out of 1% of all those bacteria, but just referring to it as a bacteridates, that's just like adding an adjective to something. That could just mean that it's previous. So the classification for that gets really messy and really hard to compare. Part of me just wants to show you guys. Maybe I should include the phylogenetic tree because it just gets really confusing really quick. With the differences in what actually constitutes what as a species. And so I think referring to each pathogen as itself is difficult enough. I think, especially since the bacteria are able to share genes across different species. If that makes sense.
So I have a very technical question. The sequencing, how do they get? Because if we don't know the bacteria, we do not have a template to compare against. We cut the DNA into little pieces, and then mix all of the species together, and then we try to reconstruct. Like if we pulled one single strand and recorded it, and then from another bacteria, another single strand, then we would at least know, oh, that's how they cluster. But we cut them at random places because that's how our the technology works, and then how I just don't understand the multi-species.
Oxford has nanopores they use. So there's these tiny little holes that only something the size, something at the nanoscale can fit through. And they usually have a filter. So you filter those through and usually get nucleotides out the other end, if that makes sense, because they're just so tiny, they're tiny holes, if that makes sense.
Well, but not entire strand, right? Because it rips off and
Yeah.
things like that. So it's still.
So you get a really big sample size, usually is what they do, just to make sure. But yeah, it's a little.
Doesn't help. Sorry, doesn't help to understand because it's still partial, right? We don't know. We still have to find another piece, right? Or we're just getting those strands and grouping them, like clustering them, regardless of the length. We're not trying to connect them first.
Yeah, that's another issue too.
So are we trying, like, in there?
We're trying to connect them. We're trying. So we feed them through the nanopores themselves, and then we just have the nucleotides that come out the other end, and that's
the sequences. So we have
yeah, and that's what we run on.
and that's it. If it got chopped off at some point, we don't reconstruct or this is the thing that we
It could just be the other 99%. That's that. I think that's one of my main critiques is that it's just yeah. Who knows? It's a false. That's why I think we end up getting a false positive. But
sounds good. Okay, well, if there are no questions, then thank you, Jordan. That's an interesting choice of a paper, anyways. And I like your introduction through the we are Jordan. We're maybe we are just engines, vehicles to transport bacteria around and feed them. That's right. That's all. What are they called? Those starships that you send to far galaxies that they go. So we're like that. How many generations of bacteria pass through in 17 years?
17.
17. Well, you have a starship, me. I'm going with another metaphor, not with our Jordan.
I like
And I'm traveling through space and time, and I'm carrying this community in me. So how many civilization cycles inside it passes? How long does a single bacterium live? So,
Depends on the species, but normally they have there's a exponential power calculation that we use in lab, I think, to estimate how fast they replicate. I can attach that if that helps.
no. Well, just off the cuff, like, what do you think? How long does it? What's like? Is it a day? A year? How long does a single bacterium in our gut live? Like a single.
Not very long. It's pretty short.
Yeah. Well, a second, a microsecond. Like, what is the scale of that short?
Depends. It depends if they're wanted or not, or if there's a immune reaction, or if there's another bacteria because they're competing too.
Yeah.
And
Like
the same space.
usually it's in the minutes range, right?
Yeah.
Minutes.
Because they go through a cycle. They go through a cycle, and they usually have binary fission where they kind of separate, and then it gets exponential. But depending on the nutrients that they have, it depends on which cycle they're in.
I don't think it's like 1 minute, but it's less than an hour.
Well, it also depends if they form an endospore, because then they're just pretty much stuck in hibernation until they come out. So, depends, I would say. But
Okay, so about 38 million minutes. So a few million generations.
over generations, you mean?
Oh, bacteria.
Yeah. Well, and then each one's going to have different mutations assorted to the environment because they're selecting towards our environment. This paper is trying really hard to combine all these different things, and I think it starts off great, and then as soon as it tries to validate it with multiple tests, it just kind of falls apart. It's my summary of it, but yeah, it's still interesting though. It's still a great read in Nature in December, but yeah.
Okay, thank you, and I'm stopping the recording.